How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, we're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA. We're all made of the same chemical.
Hello, you're listening to DNA Today, a genetics podcast and radio show. I'm your host, Kira Deneen. DNA Today informs you on what's happening in the genetic world. During my broadcast, I educate you, the public, on genetic and health topics through event coverage, news stories, book movie reviews, and interviews. Guests include genetic counselors, researchers, patient advocates, and professors in the field of genetics. Today, I'm talking about 4 events. Yes, I'm going to pack 4 events into this episode. One I recently attended, and 3 others that are upcoming, which you can attend if you're in the Yukon area. So the event that I attended was the New York Regional Cytogenetics Conference, and there were lots of presentations from people in the field of cytogenetics or that are doing research that kind of talks about cytogenetics or has implications in cytogenetics. And for those that don't remember, cytogenetics is the study of chromosomes, where DNA is packed into those 23 pairs of chromosomes or 46 chromosomes that most of us have 46. So I'm excited to share the highlights of that conference with you. I also had the opportunity to talk about the public's awareness of genetics through media, specifically social media, such as this podcast and radio show. So I'll be kind of talking about what I talked to them about. But before I get into the conference, I want to let you know about these 3 cool genetic events. 2 of them are at UConn. One of them is at UConn Health, which is about 40 minutes away from the UConn Stores campus. And what's interesting about all 3 of these events, they're coming up in the next 2 weeks, they are all about epigenetics, or at least a portion of them are about epigenetics, which really goes to show how important this aspect of genetics is becoming in research. We see it more and more in the literature publications, and not only that, but we see it more in the mainstream news when covering genetics or science. So for those that don't remember, epigenetics is the study of changes in an organism caused by modifications of gene expression, rather than the alteration of the genetic code itself. So what does that mean? It means that there are factors that influence the gene expression that aren't actually changing the code of the DNA, the ATCG, that genetic language. So it's not changing that, but it's changing what genes are being expressed. So there are epigenetic factors that are compounds that attach to or kind of mark the DNA. And these factors interact with genetic material, but they do not change the underlying DNA sequence, again, that code. So instead, they act as these chemical tags. So an example is methyl groups. So in a process called methylation, methyl groups are added to the DNA, which changes the expression level of the genes that it's being added to. And it indicates what, where, and when the gene should be turned on or expressed. And when I say expressed, I mean that gene is then turned into a protein. And an analogy I like to use for this, if people aren't that familiar with DNA and these processes that can get pretty intense, is playing in a piano. So if the genetic code is the piano, then the epigenetic factors are like the person playing the piano. So if you have a piano has keys on it. So pretend those keys are genes, and the person that's playing it is those epigenetic factors. And so when the epigenetics piano player presses on the keys, they're activating that gene, and that's what's being expressed. That sound and that note is coming out of the piano, or that protein is going to be produced. So you can have a bunch of keys there. The DNA exists, but they're not being produced into proteins. So that's kind of an analogy that's really helped me learn what epigenetics is. So the first event that I would like to talk about is the Environment, Epigenetics and Cancer, How to Cultivate the Connections. And this is an event that is going to be on the UConn campus, and it is sponsored by the UConn Center for Environmental Health and Health Promotion and the Center for Promotion of Health in the New England Workplace. And it is to recognize Breast Cancer Awareness Month. So it's going to take place on Monday, October 24th, starting at 11:30 and ending about around 2. It's going to be in the Student Union Theater on the Stores campus at UConn. And the keynote presentation is by Dr. Mary Beth Terry. She's going to be talking about the breast cancer susceptibility, rethinking the role of environmental rethinking the role of the environment and methods to improve risk assessment. So a lot about if a person is susceptible to developing breast cancer, if they have certain gene mutations, and how, aside from those gene mutations or in conjunction with, how the environment plays a role in us developing cancer, because most of cancer is not heritable, meaning that it's not inherited. It's actually from other factors than just the DNA you're born with. So there's a lot of other factors that go into that. She's going to be talking about some of that and how we can assess the risk of developing cancer through those types of venues that we're looking at. And accumulating evidence has shown that the interrelation between environmental stressors such as toxins, exposure to work hazards, and lifestyle factors as modifiers and contributors to an individual's health, specifically cancer. So the goal of this symposium is to provide attendees with an update of current research and our understanding of the role of epigenetics, which I just talked about, that piano analogy, and the environment in cancer risk and development. And so after the keynote presentation, there's going to be a roundtable panel discussion, which I always find is really great for engaging the crowd and kind of getting students, because I'm assuming it's mostly going to be students in the audience, to really participate and be able to ask questions and really be a part of that conversation. So Dr. Terry, who I mentioned before, is the keynote panelist, or giving a keynote address. She's going to be with some guest panelists, including Dr. Gary Lee Ginzenberg, who is a toxologist for the Connecticut Department of Public Health, Ellen Matloff, who is a certified genetic counselor and president of My Gene Council. And side note there, I actually work for Ellen Matloff at My Gene Council as the communication lead. So I'm excited that she's going to be there, and obviously excited to promote an event that she's going to be at and that UConn is sponsoring, because obviously I am a current UConn student. And another guest is going to be Dr. Christina Stevenson, who's an oncologist at UConn Health. And so all of them are going to discuss advances in risk assessment, implication of scientific discoveries, epigenetic biomarkers, I told you, there it is again, and technological advances in screening, diagnosing, and treatment of cancers, as well as providing feedback about the public and patient perspective. And a couple moderators are going to be Dr. Robert Henning, Dr. Judy Brown, who is actually my director of the Diagnostic Genetic Science program here at UConn. And she's also the Healthcare Genetics Professional Science Master's Degree Program director. So it's going to be an awesome event. I'm going to be there. I'm volunteering. So definitely head to the Student Union on October 24th. Registration starts at 11:30, and we're going to kick off the keynote presentation at 12, followed by the panel discussion. So I hope to see you guys there. Our next event that's going to be at UConn is actually 2 days after this, and it's called Gene Editing, the CRISPR Revolution. So this is going to be on Wednesday, October 26th at 6 p.m. in the Dodd Research Center, which, for those of you that may not be familiar with the campus, is next to the library, next to that really big book statue. So it's right there. And I've talked a lot about CRISPR on this show, talking about how it is a gene editing technology that has really taken biology and kind of just the scientific field by storm with how much ability it has and the future implications of it. So with the invention and development of this CRISPR-Cas9 technology, which is a specific type of CRISPR, it's using a Cas9 enzyme to actually do the molecular scissor cutting. Science world is rushing to develop applications and test theories for its use. It seems as if the possibilities for CRISPR are endless. It could combat issues such as drought-resistant crops, disease-resistant livestock, new drugs, and even cures for cancer. The first clinical trial using CRISPR on human cells started in China in August, and it really, the last probably 3 to 4 years, it's just become very big and very widespread in a lot of different labs. So the conference here is going to talk about how this technology works, what are some of the current explorations of this gene editing technology, what are the limitations and risks of its use, which I think is a really important one that kind of brings up that classic bioethical question of if just because we can doesn't mean we should, or should we? So that's something that I'm sure they're going to be talking about. And going along with that, what ethical conversations need to be had by science, but also the public and policymakers, that a lot of people from different areas of expertise need to come on into this discussion of CRISPR and say, where should the line be drawn? Should it be drawn? Kind of think about all of these different issues. And the Leadership Legacy Experience at UConn is hosting this event, and the keynote speaker is Sharon Beagley, the senior science writer at STAT, the life sciences publication of the Boston Globe, and she's going to discuss this invention and evolution of the CRISPR-Cas9 technology. Sharon was previously the senior health and science correspondent at Rutgers, the science editor and science columnist at Newsweek, and contributing writer at this magazine and its website, The Daily Beast. She is a co-author of the book, The Emotional Life of Your Brain. So if you can read books super fast, maybe you read her book, again, The Emotional Life of Your Brain, before the event. So again, that's going to be on Wednesday, October 26th, 6 to 8 p.m. in the Dodd Research Center. Again, these events are all free, and all members of the community are welcome to attend. You don't have to have a science background. You don't need to be a student at UConn. Anyone can come by. So the third event I'm going to talk about is the Prader-Willi syndrome, new epigenetic findings, and potential routes to therapy. Now, this is a new event that's going to be held Friday, November 4th. It's close to UConn stores, but not on its campus. It's going to be in the UConn Health campus. And Dr. Mark Lalonde is going to be presenting this. He's a HealthNet professor and chairman, and he's also in the Department of Genetics and Genomic Science at UConn Health. He's kind of everywhere. So for those that may not be aware, Prader-Willi syndrome is a genetic disorder that's usually caused by the deletion of a part of chromosome 15. So remember I talked about how we all have 46 chromosomes, or most of us have 46 chromosomes in all of our cells. So for people that have Prader-Willi syndrome, they have a deletion on part of chromosome 15. It's a relatively small deletion that can be seen cytogenetically. So if you're looking at a chromosome, you can see it. You don't need to have molecular techniques to see it, only in some cases. And this is passed down by the father, which is important because there's another syndrome called Angelman Syndrome that has the same deletion, but it is passed down from the mother. So that's a kind of whole nother conversation. But imprinting, which is kind of where you're getting your DNA from, which chromosomes, fathers have different imprinting than mothers. And so diseases can actually differ depending on where you inherited that chromosome from. The most common symptoms of Prader-Willi syndrome are behavioral problems, intellectual disability, and short stature. Hormonal symptoms include delayed puberty and constant hunger leading to obesity. That is definitely the most prominent syndrome or symptom of this syndrome, and it's very hard to miss. Someone that has Prader-Willi syndrome is really not going to go undiagnosed because this constant hunger is a huge part of their life, and they have to, parents of Prader-Willi syndrome children have to lock up food. They have to make sure that the garbage is not out and about because children will go there because they have something that's different about them that leads to this constant hunger and therefore obesity from it. Unfortunately, there's no cure for Prader-Willi, but many patients will benefit from, again, that supervised diet, and there are different kind of rehab places that can help Prader-Willi people really kind of learn this and be in an environment where food is not out all the time. So some symptoms can be treated with hormone therapy as well. So if you want to learn more about Prader-Willi syndrome, because that was definitely just kind of skimming the surface, you can attend Mark Lalonde's event, Prader-Willi syndrome, new epigenetic findings and potential routes to therapy. That again is going to be Friday, November 4th. It's going to be 2 to 3, and it's on the UConn Health campus, which is in Farmington, Connecticut. And again, all are welcome. It's going to be a free event, so definitely check it out if you're in the area. I recently attended the 2016 New York Cytogenetics Regional Meeting, which was held in Syracuse, New York, and hosted by the Cytogenetics Laboratory, SUNY Upstate Medical University. And there were lots of presentations. It was kind of a whirlwind of a day, just listening to so much research and case studies and kind of seeing what's really hot topics in the field of cytogenetics right now. And again, cytogenetics is the study of chromosomes and kind of the implications on abnormalities in those chromosomes and relating that to cancers, prenatal samples, constitutional diseases. That means you're born with them. So all kinds of different things. So I'm going to mention a few of the presentations and kind of an interesting tidbit I learned from what they were talking about. And then I'll get into kind of what I talked about during my presentation. So the first presentation of the day was Dr. Irene Cherric, and she presented a case study of ethical issues within pediatrics. And she highlighted cases of patients whose beliefs conflicted with the standard medical care. And one family she talked about was Amish, and they have a differing view of death than most people, or people kind of in the medical field. Another patient she talked about was a Jehovah's Witness who refuses blood transfusion, as most Jehovah's Witnesses do. And the important takeaway message I got from her presentations was not only, all the heavy scientific information she was talking about, but also to remember that all people, these patients are people, and that they have their own morals and beliefs. And healthcare providers really should be respectful and educate themselves on that patient's religious beliefs, or just beliefs in general. That way they can educate the patient while kind of keeping this in mind and giving them all their options for treatment. That for the Jehovah's Witness patient, for example, she had a cancer and she needed a blood transfusion, and the outlook was very poor for her to survive without one, but she didn't want one. And so Dr. Cherric, who was presenting, kind of talked about how she would always offer it to her and say, these are your options, and let her know that you have this outlook if you don't have the blood transfusion. And she continuously declined. Luckily, she did make it through and had a recovery. But I think the important kind of take-home message here is that if healthcare providers can really be educated on a patient, they can educate the patient even better, and just kind of keeping that in mind. Another presentation was by Dr. Robert Hutchinson. He presented on the updates of the WHO classification system, WHO stands for World Health Organization. And a point he made was that a genetic abnormality results in a specific disease that responds to targeted therapy. So basically what he's saying there is that even when people have the same disease, they can have different genetic abnormalities that have led to that disease. And those different abnormalities do not have the same responses to the same drugs. This is where the concept of precision medicine or personalized medicine comes into play. So subgrouping of diseases by their genetic abnormalities can increase the success of medications. Breast cancer is a great example of that there are lots of different mutations that can develop into breast cancer. And people that have that are having one type of breast cancer can differ from other people with that same subtype of breast cancer by different genetic abnormalities. And based on those genetic abnormalities, we actually give those two patients different treatments. And in some cases of breast cancer, if you give a patient a medication that maybe their peer that doesn't have the genetic abnormality, or the same genetic abnormality, I should say, if you give them that treatment, they can actually worsen. And so that's kind of a key point of having that personalized medicine. It's not, I think what the public sometimes gets confused with is personalized medicine is not every person is different. It's subgrouping further and further so that we get more precise. Another interesting presentation, Thomas Pope had an intriguing case presentation on a rare constitutional sex chromosome disorder, which I don't have the time to explain, but it was definitely very interesting. He did share two really cool fun facts, which the first of which I had no idea and hadn't heard of before, so it was kind of cool. So some women have a condition called Tetrachromacy, which is kind of like a superpower. So it's having super color vision, and it's linked to the X chromosome. Remember that typical females have two X chromosomes, typical males have an X and a Y chromosome. So this condition, tetrachromacy, is linked to the X chromosome, and it allows women to see up to 100 million shades of color. This is due to having 4 types of cone cells, remember rods and cones? And that's in their eye instead of the usual 3. So just by having 4 different types of cones, they're allowed and able to see 100 million shades of color. That kind of blew my mind. It was very interesting. And kind of turning to the male side of things, the X chromosome of all living males are related through a single male ancestor who lived over 100,000 years ago. So when we talk about passing down our genes, the X chromosome doesn't really change all that much because it's the same one. If you're a guy and you will then have the Y chromosome, from your dad, and he got that from his grandfather, and that grandfather got it from his great-grandfather, and so on, because X chromosomes are inherited either from the mother or father, so it's not really that direct line, but the X chromosome are all related through a single male ancestor who lived over 100,000 years ago. So ancestry is a really cool thing that I haven't talked about too much on the show, but I hope to in the near future because obviously genetics reveals a lot about our ancestry. Dr. Kazim R. Chohan presented on the application of molecular techniques in reproductive medicine. I found this particularly fascinating as personally I want to pursue a career in prenatal genetics. And I was really impressed when he mentioned that he helped create the technology that allowed the first baby, born just a few months ago, to have 3 biological parents. And this sounds like science fiction, but how this works is that the baby has nuclear DNA and has 46 chromosomes. 23, one set from a mother, and the other set, the rest of the 23, from the father. So that's kind of like, how we were all made, right? But mitochondrial DNA is something that exists and typically is inherited from a mother because the mother is providing kind of the first cell, is providing the egg, whereas the father is providing sperm, which is mostly just DNA. So with this child that has 3 biological parents, they have mitochondrial DNA from an additional woman. So they have nuclear DNA from a mother and a father, but they have mitochondrial DNA from a different mother. So there's 3 different adults kind of that created this child, along with all the scientists in the lab. But this was really interesting, and it really hit the media big because when people, it's kind of a very BuzzFeed type of clickbait title of, baby born with 3 biological parents. But when you look at it, it's kind of maybe not as crazy as you would have thought, but it's definitely something that is really big and that can allow families to have children without passing on mitochondrial diseases. So women who have abnormalities in their mitochondria can pass it on to their children. And they're going to pass it on to all of their children because males don't contribute to mitochondrial DNA. So kind of the fix here is don't use, the biological mother's DNA that, wants to have the child and raise them and everything like that. Use a donor's egg, but insert the mother's DNA and the father's DNA. So they're really going to have all the characteristics and, pretty much be the biological children of those 2 people, but they're going to have some little bit of mitochondrial DNA from another woman. So it's something that is very exciting for people that have mitochondrial diseases and that people that are researching this. It's definitely something that I'm going to keep an eye on and keep you guys educated on. The last presentation of the day was Dr. Frank Middleton, and he presented on the discovery of epigenetics, there it is again, and metagenomic biomarkers of autism using next-gen sequencing. So he discussed researching genetic links to autism, such as telomere length, which is the end of the chromosome, so the tips. And I kind of like to use the analogy of the aglet on your shoelaces, that end plastic part. That's kind of what telomeres are. They protect the chromosomes from degrading or losing any material. It's kind of this repeating sequence at the end of each chromosome. And telomeres have been linked to age, exercise, so many different things that are kind of broad categories. And so Dr. Middleton is looking at telomeres to see if it has any association or correlations with autism. And he's also looking at other things. One thing he talked about was the metagenomic biomarkers. And the metagenome is like an expansion of the microbiome. So when we talk about the microbiome, you should be thinking about your gut and all the bacteria in there. And it's said that we're made up of more bacteria than human cells, which is really interesting kind of to think about. But the microbiome is in the gut and kind of isolated there. Whereas the metagenome is kind of everything in our bodies that isn't human. And so that includes other bacteria, sometimes some viruses that live in our bodies. And he was measuring and looking at different levels of different components that are in saliva of kids with autism. And from doing this, he ended up learning so much about the metagenome. And this kind of shows how you start researching something and it kind of takes off in a new direction. You start discovering something new. Often this happens in basic research when we're kind of just looking for something. We don't really have something that we're trying to solve necessarily, but we're just learning for learning's sake because it could lead somewhere very interesting. And most of our advances in science have come from basic research, which kind of a random thought is that the government really needs to continuously provide money for basic research because that is where we're making so much progress and just learning about new things. So that was definitely a very interesting presentation and interesting talking about a disorder that is not cancer or not kind of a straightforward disorder. Autism is very interesting and has a spectrum, as we know, of different types of autism and how severe they are. So it was very interesting to hear from Dr. Middleton about all of that. In my presentation at the meeting, I talked about the public's awareness of genetics through social media, such as LinkedIn, Twitter, Facebook, podcasts, blogs, all kinds of different sites. And I really focused on talking about how translating genetic information is very key. And these social media sites allow science communicators, such as myself, to target lay people and the public to learn about genetics and just be educated on genetic topics such as genetic testing, hereditary cancers, all types of different topics. And what's great about social media is people are already on there. Lots of people are on Twitter, constantly checking their feeds. People are on LinkedIn looking at what their colleagues are doing. So by using social media, it's a great place to kind of catch people's attention and make them want to read about genetics. And it also forces educators to water down information into bite-sized pieces. So we're going from language that is so dense in the literature to social media language. Those are pretty much stark opposites right there. So being able to actually translate it and water the information down while still catching the important parts of it is really a huge skill. And people that have really helped the public understand about, especially cancers, for these four women I'm going to mention, are celebrities. Katie Couric, Angelina Jolie, Sandra Lee, Melissa Etheridge. There are four women who have increased the public's awareness of cancers. And they do this by sharing their story or their family member's story, in the case of Katie Couric, and really dramatically increasing the awareness of people about their specific cancers, because these women are talking about cancers, and they kind of add a relatable human angle of people like, oh, if they're talking about, say, a patient to someone that isn't so aware, if they're talking about, oh, yes, I'm positive for one of the BRCA mutations, and their friend's like, what is that? I haven't heard of that. Then the patient can say, oh, it's the gene that Angelina Jolie tested positive for and had a prophylactic surgery to prevent the development of cancer. And then people are like, oh, I did hear about that in the news. Of course, yes. So celebrities really provide perfect examples where educators can point to these people and say, this is what we're talking about. And it kind of provides more of an interest there. People want to listen and hear about it. Angelina Effect, after she wrote in the New York Times about her BRCA1 mutation status, genetic counselors and physicians saw skyrocketing numbers in calls of people wanting to learn if they had that same mutation, if they could lower their risk for ovarian cancer and breast cancer for the BRCA1 status. Something that's very key in communicating these genetic topics to people is pointing them to resources. There's only so much we can say on social media, right? There's only so much I can say in a half an hour podcast episode. So I like to point people to the National Society of Genetic Counselors if they want to learn more about genetic counseling or genetic testing. If they have a rare disorder, National Organization for Rare Disorders, which is coined as NORD. Genetics Home Reference for just about anything else. They talk about things at an easy reading level. And for students that want to learn more about genetics, the University of Utah has a fantastic resource where I started learning about genetics at learn.genetics. So by pointing to these resources, people can really learn more. And a brand new resource I want to mention is aboutgeneticcounselors.com. That's also from the National Society of Genetic Counselors, talking about kind of the basics of genetic counseling, and they have a section on there geared towards students. So that's a little bit about what I got to talk about during the meeting, but I am out of time for the show. So thank you so much for listening, and you can go to dnapodcast.com for all episodes. It's also a brand new website, so definitely check it out. It's not the ancient website I had previously. Let me know what you think, too. You can tweet at DNA Podcast. I'm on there a lot. And any questions or feedback or comments, anything like that, info@dnapodcast.com. Thanks for listening, and join me next week to learn and discover new advances in the world of genetics.